We are now going to talk about the control of microbial growth. Chapter 13. The CDC classifies infectious agents into biosafety levels based on their potential risk to laboratory personnel and the community. Each level has specific controls for the containment of these microbes and these biological agents. The risk level is determined based on their infectivity, how severe the disease is, how easily it can be transmitted, and what is the type of work that is being done? Are you developing a vaccine? Are you figuring out disinfectants that can control this? Are you using PCR to make more of it? Each level has specific things that must be done, specific PPE that must be worn, specific things that must be done for the protection of the laboratory personnel. The control of microbial growth uses methods that are used to destroy, remove, or inhibit the growth of microbes. It can be on surfaces, in the environment, or on the body. Most of these approaches are non-selective, which means they can control the growth of a wide range of microbes. You can have physical methods, you can have chemical methods to control microbial growth. Simple hand washing with soap is still considered the most important step in preventing the spread of many microbes, and this does not have to be antibacterial soap. As long as you are scrubbing your hands for at least 20 seconds and following the recommendations of drying your hands after you wash them, it is very, very effective. In 2016, the FDA actually banned many of the antibacterial ingredients that were being used at the time. They were concerned about the long-term effects of these antibacterial ingredients, and also the industry that was using these antibacterial ingredients hadn't proven that they were better than just washing with soap and water. Here are some definitions that you will need to memorize. Sepsis, when it pertains to the control of microbial growth, refers to microbial contamination. It means that microbes are present. Asepsis means the absence of significant contamination, without contamination. Sterilization, when we are talking about the control of microbial growth, means the removal of all microbes. If something is sterile, that means it has had all of the microbes removed. Commercial sterilization is specific to food. It seeks to destroy the microbes that can cause foodborne illnesses, the microbes that can have endospores that can come back to life when they are heated. Those things include things like C. botulinum. That is one bacteria that they are really seeking to remove from your food through commercial sterilization. Disinfection is the removal of pathogens. Antisepsis is the removal of pathogens from living tissue. Notice that asepsis is different from antisepsis. You have to look at these definitions and find the key characteristics of each of these definitions. Find those differences so that you will be able to recognize them on quizzes. Continuing with more terminology, we have degerming. Degerming is removal of microbes from a limited area. That's just removing them. That's all it is, and it's from a specifically small area. Sanitization is lowering microbial counts on eating utensils so that it is acceptable to eat from. So, some of you might have a sanitization button on your dishwasher, and when you push that, it specifically sanitizes your dishes and your eating utensils in your dishwasher. So sanitization for our purposes is specific to eating utensils. If you go into a restaurant and you see a spoon that has something on it, do you feel it's acceptable to eat off of it? If not, then to you, that has not been sanitized. But if you go into a restaurant and the eating utensils are all sparkly clean and you look at them and you go, yep, those are sanitized, I'll eat off of those, then that's how sanitization works. Either it's good and acceptable to you or not. Then we have biocide or germicide. When it ends in side, it means that you're killing something. And in this case, it is specific to killing microbes. Then we have bacteriostasis, which I will talk a lot about in this class. It's inhibiting, not killing microbes. So you are inhibiting its replication, or maybe you're inhibiting something about that microbe, so it kind of puts it on a pause. It doesn't kill them, it just stops them from replicating or stops them from growing. You do something to inhibit something about that microorganism. This is a really nice table from your book, and it breaks down some of those definitions that we just talked about, giving you a little bit more information on each of them. Let's go through a few questions. You make a peanut butter and jelly sandwich, and the knife with the peanut butter on it goes through your home dishwasher cycle. After the dishwasher is finished, the knife still has peanut butter residue on there. Is it A, sterile, or B, not sterile? The answer is B. The knife is not sterile, and it's not sterile for a number of reasons. You have to have a very high temperature and high pressure and water all combined in order to sterilize eating utensils like this. Now, if you had a commercial dishwasher, you would probably be able to get to those higher temperatures in order to get rid of all the microbes. Remember, sterile is removal of all microbes, but your home dishwasher is not likely to get to a high enough temperature with a high enough pressure and have the water all combined in there. It is pretty good and it is keeping you safe, but it is not likely to be sterile. You will have removed many of the microbes, right? You're degerming it for sure, but sterilization, most likely not. Now remember that knife still had the peanut butter residue on it after it went through your dishwasher cycle. Is the knife sanitized? A, yes, or B, no? Probably most of you would say no. The knife is not sanitized. if you pulled out a knife from the dishwasher and you saw the peanut butter still on there, you probably would want to wash it by hand or put it through the dishwasher cycle again on the next time you're washing your dishes, because you would find that it is not acceptable to eat off of. Remember, sanitization is specific to eating utensils, and it is creating an environment so that it's sanitized, that you are making it acceptable to eat from. Now, some dishwashers are pretty fancy. I don't have one like that, but a lot of you might, that actually have a sanitizing button. And if it does have that sanitizing button and you took the silverware out, then it would be sanitized. Understanding nosocomial infections is very important for this class. It's also very important for you if you are going to be going into any healthcare fields. Nosocomial infections are hospital-acquired infections. They are not the initial infection that someone goes into the hospital for. It is a secondary infection. A hospital-acquired infection was not present when they went in. They acquired it while they were in the hospital. Say someone goes in for a heart surgery and they end up with a urinary tract infection. That would be a hospital-acquired infection. When a patient goes into the hospital, they are very vulnerable. Their immune systems may not be at their strongest. They have a reduced immune system. They are in a weakened condition. They may be under great stress. They may have comorbidities, which means they have additional factors influencing their health. They may be going in for a leg wound, but they also have heart disease or diabetes, and that may also complicate things. They may be going in for invasive procedures. These invasive procedures will be a portal of entry for microbes. There are many things that can influence how someone acquires a nosocomial infection. You also have to consider that there is a higher than normal density of individuals with infectious diseases in one place. There are a lot of people in this hospital that may have an infection, and someone going into the hospital may be put in a situation where many people around them have an infectious disease. You also need to consider that a hospital environment is super cleaned. And if you think about that, if you are always super cleaning the surfaces in an area, what you're doing is you're getting rid of the weaker microbes that might be present. But then the stronger microbes might become more active, and those are the ones that are left. Yes, you get rid of the weaker ones, but the stronger ones are more likely to be present in a super cleaned environment. This chapter on the control of microbial growth has a lot of definitions that you need to understand. So let's talk about a few items and what that means to microbial growth and also to be considered for the control of microbial growth. Critical items penetrate the body tissues, like a hypodermic needle, something that pierces the skin and can penetrate the tissues. A scalpel, something that will make a portal of entry, is a critical item. A semi-critical item comes into contact with, but does not penetrate mucous membranes. So, That would be something like an oral thermometer. It touches the mucous membranes of your mouth, but it does not pierce them. It does not go through the tissues. Non-critical items come into contact with unbroken skin. That would be like a stethoscope or a blood pressure cuff. You can touch the surface of the skin, but it's not penetrating the body tissues, and it's not touching any mucous membranes. A forehead thermometer that touches your skin. It does not come into contact with mucous membranes, and it does not penetrate the body tissues. Those types of things are non-critical items. Considering those definitions, then, a hypodermic needle is A, critical item, B, semi-critical item, or C, non-critical item. The answer is A. It is a critical item. It penetrates the body tissues. It creates a portal of entry for microbes. A urinary catheter is A, critical item, B, a semi-critical item, C, a non-critical item. The answer is B. A urinary catheter is a semi-critical item. It does not penetrate the body tissues if it is a urinary catheter, but it is touching your mucous membranes of your urethra. A stethoscope is A, critical item, B, semi-critical item, C, non-critical item. The answer is a stethoscope is a C, non-critical item. You just touch the surface of the skin. You don't touch the mucous membranes with a stethoscope, and you do not pierce the body tissues. A cartilage piercing is A, critical item, B, semi-critical item, C, non-critical item. The answer is A. It is a critical item. You are penetrating the body tissues. You are penetrating the tissues. You are piercing the cartilage. You are creating a portal of entry for microbes into that cartilage area. That's often why it's very difficult for some cartilage piercings to heal, because it is a critical item and you have pierced the body tissues. Remember in the lab how we want to have good aseptic technique? We do not want to introduce any additional microorganisms into our cultures or bacterial samples. We want to have a pure culture. So in order to work with pure cultures and to have good aseptic technique, all media and instruments that contact the culture must first be sterilized. So the media would be like agar or brass for us, and our instruments would be like our loop. We sterilize our loop in the Bunsen burner flame until it's red hot, burning off all microbes that are present on that loop. We are sterilizing that loop. We are removing all the microbes. Also, when we are done with our samples, when we are done with our cultures, when we are done with our broths, all of these materials used to grow the microorganisms must again be treated before disposal. They have to be sterilized before disposal because we do not want to contaminate the workers, the people who are doing the disposing of our microbes, and we do not want to contaminate the environment in any way. Now, remember those biosafety levels of 1 to 4 that the CDC recommends? It's very important that people who are working with pathogenic organisms have even stricter precautions than we have in our lab. You can imagine someone who's working with COVID-19 and the strict precautions and the high biosafety level that must occur to protect the people who are developing the vaccines or developing the treatments, developing the antibiotics. There have to be strict precautions of safety. Now, there are some problematic microorganisms that we have to be even more careful with. We have to think about the microorganisms, the bacteria that can form endospores. The bacteria like Bacillus or Clostridium. Those types of bacteria are highly heat and chemical resistant. We have to be very careful with those bacteria that form endospores. We also have to be careful with the Mycobacterium. Remember, Mycobacterium that we identify through acid-fast stains, we have to be very careful with because they're highly chemical treatment resistant. They have that mycolic acid in their cell walls. Pseudomonas, which we've mentioned before, can grow in disinfectant. It can grow in bleach. Pseudomonas can cause some very serious infections, especially in those who are immunocompromised, those who have burns, and it can cause many nosocomial hospital-acquired infections. We talked a little bit about naked viruses before. Remember, those are the viruses that lack an envelope. And we talked about how you would think if it had an envelope that those would be the really resistant ones. But in fact, it's the naked viruses. Because they do not have a lipid membrane, which can be attacked by alcohol or disinfectants, they are resistant to chemical treatments. They can survive a long time on surfaces because they lack that lipid target. Let's consider the exponential death in the control of microbes. Decimal reduction time is the time it takes to kill 90% of the population of microbes. How long does it take for that bleach to be effective? How long does it take for that disinfectant to work? Have you ever looked on the bottle of bleach or a disinfectant that you're using, a bathroom cleaner, whatever it is? On the back, it gives you a recommended time that you need to leave that chemical on. Those times are the decimal reduction times. How long it takes for that chemical to kill 90% of the population of microbes. If it says you need to leave it on for 5 minutes and you spray it on and immediately wipe it off, you haven't been effective in your cleaning. You have not killed the number of microbes that disinfectant says that it can kill. You have to completely clean the way a disinfectant tells you to. However it tells you to, however long you need to keep that on there, you need to follow the instructions. Effectiveness of antimicrobial treatments depend on a few things that you need to know. It depends on the quantity of the microbes that are present. How large of an amount of microbes is there? It takes more time to kill a larger population of bacteria than it would to kill just a few bacteria. If you are disinfecting a patient's room who was very, very sick, if there is a lot of waste present or a lot of body fluids present, it will take a lot longer to clean that particular room. There are more microbes present. Environmental conditions also affect the treatment. What is the temperature? Is it too hot? Is it too cold? How much debris is there? Is it dirty? Is there a lot of substance there and less liquid? You have to consider how many bacteria are present and how much of it. Like, is it chunky? You have to consider the amount of debris. Biofilms have to also be considered. Biofilms, remember, are those encased communities of bacteria that work together to resist disinfectants or resist antibiotics. That time of exposure is very important. And you have to consider the microbial characteristics. What are their virulence factors? Do they have a capsule? Do they have an endospore? Do they have flagella? What do they have that can make them more virulent, more likely to be pathogenic? All of these things combine to determine the effectiveness of an antimicrobial treatment. Higher temperatures can result in faster disinfection, but too high can reduce it. And remember, we should also consider, is it a thermophile or a psychrophile? You have to consider the microbe itself. And again, dirt and organic compounds can interfere with disinfection. You cannot just spray over some chunky mess and think that that disinfectant is going to be able to penetrate it. You have to remove the organic compound, the dirt, the debris, and then disinfect. Hardness of water needs to be analyzed. It can reduce decimal reduction time, the amount of time that it takes to kill whatever microbe it is. 90% of those microbes, remember. Relative humidity in the atmosphere. Is it raining out? Is it summer? Those types of things can also infect disinfection. And there are always exceptions. Bacteria have a lot of resistance factors, and you have to know that disinfection may not always work depending on what it is. Methods that control microbial growth have different antimicrobial actions. They do different things. They target the bacteria or the viruses in different ways. They can alter membrane permeability. They could damage the proteins. They could damage the cell wall in some way. They can damage the nucleic acids within the cell. They can damage the cell so that it does not have that ability to replicate. It can affect the life cycle of that microbe in some way. Disinfectants and antibiotics and treatments for viruses are developed to have different types of targets, and they have different methods of actions. This is a table that can help you to understand different types of methods of using heat to control microbial growth. So boiling is not reliable for killing endospores because boiling can actually produce endospores. But boiling, if it's done for an extended period of time, can destroy many microbes that are present. Sometimes when people are backpacking or on extended camping trips, they might have to try and boil the water that they find in order to try and kill the microbes within that water so that it becomes drinkable. It's not always a reliable way to make sure that your water is drinkable. So there are other methods that you can do now when you are backpacking. But these used to be the standard way that you would do things to get rid of microbes from water. Pasteurization is pretty specific to food like milk or juices. When you go to a farm stand, you want to make sure that the juices that you're buying is pasteurized. It can decrease the number of heat-sensitive microbes. So it can include getting rid of some of the microbes that can cause spoiling of that particular thing. Your milk will last longer, your juices will last longer in the refrigerator. It's also helpful to get rid of pathogens within milk and juices that could potentially make you sick. Pressurized steam can be used. I'm going to talk a little bit more about how pressurized steam can be effective. It's often used for sterilizing surgical instruments. You can also use it for canned food purposes. Dry heat is what we use in the lab. We can use a Bunsen burner flame and we can sterilize our loops before we take a sample in order to culture it. So using things like a fire or a flame, that would be dry heat. There are different types of filters that have been made to help to reduce microorganisms that might be in water or might be in the air. You've probably heard of HEPA filters. HEPA filters are high efficiency particulate air filters. That's a really long sentence. That's why it's shortened to HEPA. Now, HEPA filters can be found in your vacuum. You can also have portable standing air purifiers that have HEPA filters. You even have HEPA filters now in some cars to help remove some of the microorganisms that might be in the air. When I was talking about boiling before for water when you're backpacking or where you're in areas where you might want to try and remove the microorganisms from the water that you have, they now have developed something really cool called a life straw. And this life straw can be used for backpacking and extended camping trips, or for areas where water is not the best and you want to try and filter out some of the microorganisms. This life straw can actually filter out some bacteria, parasites, and microplastics to make it safer for you to drink. Some of these methods for controlling microbial growth might start to make sense to you now. These are some more physical methods for controlling microbial growth. We can use low temperatures like refrigeration or deep freezing, snap freezing, or freeze drying to control microbial growth. Think about your refrigerator. You are controlling the growth of bacteria in your food. You are preventing the food from spoiling through this growth of bacteria. And what type of bacteria are you controlling? You're controlling the thermophiles and the hyperthermophiles that love heat, right? You are controlling the bacteria that don't like to be in these very cold temperatures. You can use high pressure. You can crush the microorganisms and denature their proteins. You can use desiccation or dehydration to remove the moisture. This will prevent metabolism. And you can use osmotic pressure. Remember, that's the influence of salt or the influence of sugar, those concentration levels. The higher the concentration of salt, the higher the concentration of sugar, the higher the osmotic pressure. And that can cause plasmolysis. You're affecting osmosis. Would you eat radiated food? Yes or no? If you eat processed foods, things that are shelf stable, you likely eat radiated food because they use radiation to control the growth of microbes in those foods, and they can last a long time on the shelf. Those Twinkies can last a very long time compared to a nice sponge cake that you make by hand with simple ingredients from your pantry. We don't use radiation, and our foods that we make may spoil quicker than those that are on the shelf that have been radiated and the shelf life has been increased. They are preventing microbes from growing in these processed foods through the use of radiation. The CDC reports up to half of all Americans say they would not eat food that has been irradiated. No radiation has been used on that food. But they don't know that if they eat processed foods, that radiation has likely been used to control microbial growth and make it more shelf stable to make it last a whole lot longer. Now, radiation damages the DNA of a microbe. It prevents it from growing. It prevents it from replicating. There are many different types of radiation that can be used. Ionizing radiation, which includes X-rays, gamma rays, electron beams. There's non-ionizing radiation, UV light. Now, UV light is not just used for things like processed foods to control microbial growth, and we will talk about that in a minute. Microwaves, you might think, oh, that's using radiation. Actually, what it's doing is it's killing by heat. It boils the water or the liquid material in that food. UV light can be used for disinfection of surfaces. UV radiation causes the formation of these dimers in the DNA. It leads to these lethal mutations in these microbes. There are many different types of wands and lamps and things that are used that can emit UV light. They're often used in laboratories to sterilize different types of equipment. So yes, UV light has been used to control and disinfect surfaces. Again, the effectiveness of disinfectants requires certain things. You must use the proper amount, the concentration of the disinfectant in the amount that the instructions say needs to be used. It needs to be put on for the recommended amount of time for that decimal reduction time to occur. 90% of the microbes will be killed, but you have to use it for the required amount of time. It needs to be free of debris and organic matter. You can't spray it through the chunks and think that it's going to disinfect the surface. You have to remove the debris and then disinfect. The pH also matters. So there are things that are required to have effective disinfection. An old school method of evaluating the effectiveness of a disinfectant is the dilution test. You take these metal rings and you dip them in whatever bacteria you are trying to see if a disinfectant works on. So you dip it in the test bacteria and then you let it dry. And then you take that ring with the dried bacteria on it and you place it in disinfectant for 10 minutes. Then, after you have done that, you kind of stamp it on a culture media. You use it like a stamp and you just kind of touch it onto the culture media, say like a petri dish with agar. So, by doing that, you can see how much bacteria grows from that ring because you've dipped it in bacteria and then you dipped it in disinfectant, so hopefully you killed a lot of the bacteria, but you are seeing if it will grow on the petri dish, on that agar. You want to see how much survives that particular type of disinfectant. Today, and in fact, in our lab, we're going to do a disc diffusion method. We are going to test antibiotics in the lab. We are going to see which ones are effective for which bacteria. It's called the disc diffusion method, and it uses a special type of agar that helps whatever you soak that disc in, it will disperse throughout the agar. It's easy, it's more quantitative, it's a really excellent way to test disinfectants or antibiotics. When we look at disc diffusion, we are first going to make a lawn of bacteria on a petri dish. And then we are going to place the soaked paper discs onto that lawn of bacteria. They've been soaked with an antimicrobial of some type. They have been soaked with an antibiotic or a disinfectant. Whatever you are evaluating is on that disc. When you tap that disc onto that petri plate, you then incubate it. And then you can measure what's called the zone of inhibition, where no growth of that bacteria occurred because whatever it is you are evaluating affected the growth. The bigger the zone of inhibition, the bigger the zone of no growth, the better that antimicrobial is. Little zones are not as effective as the larger zones. If you look at A and you look at number 1, that is a smaller zone of inhibition than if you look at number 5. You can see which one would be better based on that zone of inhibition when you are using the disc diffusion method. Here are a few chemical compounds that are used for disinfection purposes and control of microbial growth. You can see the A, B, and C. Those are the chemical structures of these compounds. Some of them have been around for a really long time, like carbolic acid. Lister used this. Lister sounds like Listerine. We're going to talk about that in a minute. But carbolic acid, which is a phenol, can irritate the skin. Phenolics, which are like phenols, like Lysol. Lysol is a phenolic. It's in soaps, it's in detergents, it's in mouthwashes, it's in throat lozenges. It injures the plasma membrane. These different types of chemicals affect microbes in different ways. You might have heard of BPA, bisphenol A, and the removal of BPA from plastics. BPA was put into plastics for years. When Tupperware was invented, it had this chemical compound in the plastic. It made the plastic pliable. It prevented it from shattering when it was dropped on the kitchen floor. And the whole key about the BPA was that it helps to control microbial growth. It helped to aid in the prevention of the spoilage of your food when you put it in this plastic container and you put it into the refrigerator. It also helped. The problem is that BPA can leak from the plastic into the food that's contained within that plastic. And BPA has been found to cause neurological problems and many health issues. And so BPA was removed from many, many plastics that are on the market, the majority of the plastics. There is also something called triclosan, and it has now been incorporated into kitchen plastics. But it is also something that was used in soaps, but it was removed. Remember how I talked about how some antibacterial ingredients were removed in 2016? This is one of them that was taken out of antibacterial products because they were worried about the long-term effects. Now let's revisit plastic for a second. Have you ever kept your water bottle, your plastic water bottles that you shouldn't be using, but a lot of people do. They get them from the grocery store. Maybe they use them for sporting events. They have plastic water bottles. Do not leave your plastic water bottle that you bought from the store in your car or in an area that it can get warm, because the chemical compounds from that plastic can get into your water. Have you also ever noticed that there's an expiration date on the water bottle? It's not because the water expires. It's because the plastic is made with chemicals that can leak into the water over time, and you shouldn't drink that water past the expiration date. Now, these types of chemicals they were used with good intention because they want to protect us from microbes or eating food that can make us sick because it's spoiled. So there are good intentions in all of these different types of disinfections. You just need to be aware of these different types and methods of disinfection and what that could mean to your health, and whether it's something that you want to use because you do feel it's effective and it is beneficial for you. When we hear about hand sanitizer, there has been a lot of talk about the percentage of alcohol that is in that particular hand sanitizer or in that particular type of disinfectant. The key for alcohol being effective is it cannot be 100% alcohol. It has to have some water in order for it to work, because it utilizes osmosis and the flow of water to destroy the cells. Also, when you're considering hand sanitizer, remember that alcohol portion of that hand sanitizer is going to destroy the lipid content or the lipid membrane of whatever it is that it is being used for whichever type of bacteria. So, alcohol is also ethanol. It denatures proteins. It dissolves lipids. And remember, it cannot be all alcohol. The best percentage is around 70%. Heavy metals can also be used to control microbial growth. Silver can be combined with antibiotics to increase the effectiveness of antibiotics. Mercury has been used previously, but it has very serious toxic side effects in the kidneys and in the central nervous system. So mercury is used very sparingly now. Copper has been used to kill algae in swimming pools and fish tanks. You can line incubators with copper so that when you put in your bacteria cultures, that the bacterial cultures will not grow on the walls or the floor of the incubator. metals bind to the proteins and they can inhibit the enzymatic activity of that microorganism. It will denature the proteins. If you disrupt that protein synthesis, then microorganisms are not able to continue to grow. And so that's how the heavy metals would work. You can see in the images below that in A, you can incorporate heavy metals into door handles or materials where you touch them and can be transferring microorganisms. There are some handles and metal pieces on campus that have been treated and resist microbial growth. Silverware, true silverware was developed to help control microbial growth. And again, about the incubators, you can line them. Listerine, a lot of times zinc is used to help when you are trying to control microbial growth. And zinc is added to a lot of things like Listerine and a lot of different things like throat lozenges. Now, the gentleman in E, there's something that some people take to try and boost their immune system and protect themselves from microorganisms. Some people will ingest silver. It's called colloidal silver. But if you ingest too much colloidal silver, you can turn a permanent color of the shade of blue. And that's what happened to the gentleman in E. He ingested too much colloidal silver over an extended period of time. Now, a little tiny bit is okay, but if you take it continuously in larger amounts, it is not very good for you, and it can turn you a permanent color of Smurf blue. Here is an example of copper being used in bed rails. So again, in a hospital situation, if you could incorporate heavy metals into bed rails or doorknobs or line air ducts, that would be really beneficial for controlling the spread of microorganisms. If we had these really cool metal lab tables, then we would be able to control the spread of microorganisms in our labs. And again, keeping our incubators from spreading when we're trying to grow cultures. We don't want it to spread on the walls or on the bottom of an incubator. Here is a table of surfactants, surface active agents that are used for the control of microbial growth. Soap can be a surfactant and it's used for degerming. Let's look at that word again. It's the removal of microbes from a limited area. It's not sterilization, it's degerming. Acid anionic detergents are for sanitizing. It's something that you would use in your dishwasher. We have quats, which we are going to talk about on the next slide. They're bactericidal. They denature the proteins. They can disrupt the plasma membranes. And iodine can be used for degerming. This image is from your textbook, and it represents the structure of quats. And you might recognize that bottle of Bactine. A lot of people have Bactine in their first aid kits at home or in their cabinet at home. And it's used for minor scrapes or scratches or cuts. It helps to prevent infection. That quat, remember, disrupts the plasma membrane of the bacteria. It reduces its integrity, its strength, and it causes the death of that bacteria. So now you know how Bactine works. Here are some examples of some iodine products or iodine-based products. Remember, iodine type of disinfection is for degerming. In addition to radiation, Chemicals are added to food for food preservation, to protect us from food spoilage, to control the growth of molds, to control the growth of bacteria in our foods. Organic acids are a very common food preservative that you might have seen when you've read an ingredient list of something that you are eating. You might have seen the words sorbic acid or benzoic acid or calcium propionate. These are all organic acids that help to prevent our food from spoiling. Nitrates is another chemical that has been added to bacon or lunch meat or hot dogs. Will often have nitrates in them. Many organizations have encouraged people who produce hot dogs to remove these nitrates, these chemicals, because some research has shown that they can morph into cancer-causing molecules. Antibiotics like niacin or netamycin are used to prevent the spoilage of dairy products or meats. We are going to talk about the problem with the use of antibiotics in animal husbandry. When an animal is injected or antibiotics are used for the prevention of spoilage of meat or for the growth of that animal. For us to ingest that low level of antibiotic over a period of time can make us more resistant, and we will talk about that coming up. So while antibiotic use is used in food preservation and it can protect us, we also need to think about the consequences of prolonged low exposure to antibiotics in our food. Using compression to control microbial growth is a food preservation technique that they are working on for use in the future. They use extreme compression at 100,000 psi. They've been working on it at OSU, and unfortunately what happens, even though it gets rid of all of the microorganisms, it turns whatever food they are treating into a mushy consistency. And most people do not want to eat a mushy type of food. That texture is something that many people do not like. However, they could use this compression technique for things like baby food because it's already a soft type of food, or dog and cat food, the canned cat food or the canned dog food. They could use compression preservation instead of the introduction of chemicals or radiation into these types of foods. They can prevent the growth of bacteria and make it mushy, and dogs and cats won't mind and babies won't mind. So if you are a vegan or a vegetarian, this might be something that's very important to you. You should look into this next food preservation technique that I want to talk about. So I didn't know this, and I was a vegetarian for years and a vegan, and I did not know that many bananas are treated with chitosan. So chitosan is made from crushed crab shells and lysozymes. But that is a type of seafood, and they coat these bananas with this chitosan because it's antimicrobial. It prevents the bananas from spoiling. It slows down the browning of bananas. So these bananas can be shipped from very far away with this chitosan treatment on the outside from crushed crab shells to treat it, and it can protect these bananas so they don't turn brown in transit and they last longer on your counter. It's something that you should be aware of and maybe look into if this is a concern of yours because you are a vegan or vegetarian. Let's look at this table of common agents that are used to control microbial growth. You have those phenolics, the quats, the chlorines, the alcohols. If you look at this list at this table, you can see that no one particular type of agent is good at all things. Nothing has been found that is effective against endospores and effective against mycobacteria. They can kind of do one or kind of do the other or none at all. None of them is the perfect type of disinfectant. You have to use disinfectants for different reasons. And again, you have to follow the instructions of that disinfectant and use it properly. These are the 5 steps for hand washing that the CDC recommends. You need to be able to state all 5 steps and to say what is happening in each step. Make sure you include the details for each step of hand washing. Hand washing can be for the general public like this, with the 5 steps for scrubbing for 20 seconds included within that. Or you can also have surgical scrubbing. Surgical scrubbing is much more extensive, and it's used for the personnel that work in the OR. But for general public hand washing, you need to know these steps.